Hey, it's great to see all of you today. My name is Mike Angelo. Really happy to have you guys for our third annual spatial biology workshop. Well, now it's a summit. It's a summit. We've steadily ratcheted up the language that we use to describe it to emphasize the breadth and comprehensiveness of this conference. Isn't that right, Sean? So
I think it was because someone said it didn't feel like much of a workshop.
that's true. That's true. We've kind of moved away from workshop territory. And so, no, I'm really glad that everybody could be here. We've got a ton. Actually, I think we had close to 200 people that are registered to be here on site, and around 100 that are joining us remote. So I think it's going to be a really great opportunity to hear from some of the premier labs that are doing work in this area from around the country, and also an opportunity, hopefully, to strike up talks and maybe new collaborations and stuff like that. Candace, I said yesterday to Candace, who is one of the organizers, that I said, "Oh, I'm just going to get up and just say a few words. I don't think I'll have any slides." And then when I got on Slack this morning, I saw that she had made me some slides. So I'm going to kind of, so can't take credit for these, but she very nicely made a QR code if you want to download the agenda or anything like that. Everybody, you got 5 more seconds to look at it with your camera. Okay, so this is pretty bonkers. She went and she made this wonderful map to show where we have people from over 98 institutions. Don't want to be inaccurate there, Candace. So 98 institutions, 14 countries, and 24 states this year. Want to remind you guys about the poster session tomorrow night. We had a lot of really great posters, almost 50 posters from grad students and postdocs and other folks, which we're really excited to be able to have everybody see. I think, is there drinks or anything at the poster session? Oh, it says it. Light appetizers and drinks. You can tell I didn't make these. And happy hour tonight. Yeah, that's right. So anybody that wants to join us at the Rose and Crown, we'd love to see you. That's from 5 to 7 p.m. It's in downtown Palo Alto. The other thing is I wanted to be sure, Candace did not put this slide in. I had to put this in. Our 3 conference organizers, which really did almost everything in terms of organization, getting sponsors to put in money so that we could, it might not be obvious, but I won't go into how much, but having this room at Stanford costs way more than it logically should. And so the registration fees alone we wouldn't be able to cover the conference. And so it's very glad that our team was able to bring in money from NIH through HubMap and also through our sponsors, which I'll show you in the next page. But before that, I wanted to give Cameron and Candace and Davide a big round of applause for So yeah, thank you guys. And Candace, thank you for, I think it was totally on brand that you did not put in an acknowledgement slide for yourself in there. So, but we also wanted to be sure to, call out our sponsors and thank you for donating the money to help us put on this conference. Glencoe, RareSite, Naval Medicine, 10x Genomics, Lunaphore, Abcam, Alpine Glow, Akoya. And also a very special thank you to HubMap, who I think for the 2nd or 3rd year in a row has, third year, has put up a substantial amount of money to help us put this on, and also to support trainee travel and stuff like that. They're very big proponents of making sure that we get grad students and postdocs involved as much as possible. So thank you for that. And so now, without further ado, because I don't really have anything cool to say, I'm going to get this first applications of spatial biology session started. And we're very lucky today to have a newly minted PI from the NIH, Erin McCaffrey, who is also the principal investigator of the NIH Spatial Immunology Unit. And she is going to talk to us about her work looking at TB granulomas, which I think is really cool. So I'm looking forward to hearing it.
All right, good morning. Thank you, Mike, for the introduction, and thank you guys for being here early to hear me talk about TB granuloma. So as Mike said, my name is Erin. I actually was Mike's first graduate student, so Mike indoctrinated me in all the ways of spatial immunology, and I'm excited to share with you today my applications in that area, specifically in an infectious disease setting called tuberculosis or TB. And so TB, as I mentioned, is an infectious disease. However, when you compare it to other infectious diseases, especially those of the lung, it has quite a distinct pathogenesis. It's caused by a bacterial organism known as Mycobacterium tuberculosis, or MTB, that's inhaled into the lung where it takes up residence in macrophages. However, instead of this initiating a conventional immune response that would lead to the degradation of the pathogen, presentation of antigen, and subsequent clearance, TB actually has a number of different virulent strategies that evade these processes. And instead, this leads to a chronic and structured form of inflammation known as granulomas at the site of infection. And granulomas are what I describe here as an immunological paradox, because in terms of the diversity of immune cells and the magnitude of response, there's certainly nothing lacking with the immune response to the pathogen. However, granulomas actually often become the site of long-term bacterial persistence and dissemination. And in many ways, they've also been described as bacterial tumors because of the ways in which that TB granulomas can spread and disseminate to other organ sites in the body and cause damage to the host, in addition to benefiting the pathogen's persistence. Now, something that's really notable about granulomas and also speaks to the similarity of this process to something like tumorigenesis is that a single individual can have multiple granulomas that form based on multiple sites of bacteria being phagocytosed by macrophages. However, within the same individual, even in the same lobe of the lung, some granulomas actually can successfully sterilize infection, while an adjacent granuloma can actually have a very disparate outcome and instead become the site of bacterial persistence, over immune pathology, and ultimately dissemination. And so this really speaks to the fact that the mechanisms dictating outcome in the immune response to TB are very locally regulated within granulomas. Furthermore, and I think this is a theme that's going to come up again and again throughout the next few days, is that if we really want to understand what's making the difference between one of these so-called good granulomas that is sterilizing versus a bad granuloma that is permissive to the bacteria, we really need to understand not just the composition and state of cells within the tissue, but also the way in which those cells are organizing and interacting with each other in a way that might indicate some spatially coordinated biological response. And our goal with this work is really to understand these features of granulomas across this spectrum or diversity of outcome so we can identify the factors that do allow the host to overcome infection and use those pathways to think about designing things like host-directed immune therapies or improved vaccine strategies. Now, before I dive into some of the data, I wanted to give a bit of a roadmap to some basic attributes of granuloma structure that will become relevant both to the way we think about the immune response in this setting, but also in the way we think about computationally analyzing the data we get from these tissues. And so this is that granulomas are intrinsically compartmentalized. What I mean by that is that granulomas have this consistent recurring structure where they have two primary zones. The first of these we called the myeloid core. This is the center of the granuloma that's predominantly made up of different macrophages and monocytes. This is also the hub of infection. It oftentimes takes on the appearance of central necrosis or cell death. And this is importantly that hub of infection. So the reservoir of bacteria in a granuloma will be found in this environment. Now, on the outside of the myeloid core is a second zone known as the lymphocytic cuff. This is an environment predominantly made up by T cells and some B cells. And what's notable about this environment is that these lymphocytes encircle the myeloid core, but they largely actually fail to infiltrate within the myeloid core. And this has been an attribute of granulomas that's been long known since the first pathological descriptions of granulomas in the 1800s. However, what leads to this compartmentalization is not well understood, but it's thought to be quite consequential that lymphocytes, which of course are critical for adaptive immunity, are not able to successfully infiltrate into that hub of infection. You might think of this in the same way you think of a tumor that's poorly infiltrated by immune cells. And so keep these features in mind as we start to walk through some of the data. Now, a couple of years ago, we actually did take a first stab at trying to understand the basis of both immune cell diversity, but also why granulomas in human TB disease often fail to control infection. And this was a study we did using a multiplex imaging technique developed here at Stanford by Mike Angelo called MIBI that essentially allowed us to take a set of human clinical tissues from patients who had TB and look at the distribution of both 37 proteins and 19 types of cells within the tissue. And I'm going to highlight this study for you just in 4 broad strokes about what the really important takeaways were that gave rise to the set of work that we've been doing today. So, as I mentioned, granulomas are often a site of failed immunity, but largely we don't understand why. There had been some thought in the field that this was due to dysfunctional Th1 or interferon gamma derived T cell responses, but largely this model was derived from murine models of TB that we know don't model the disease process well. And so we fundamentally had questions about what factors or attributes of the granuloma might suggest to us that this environment is not poised to control infection. And in this way, a lot of our data came back to those similarities with the tumor immune microenvironment, where we found that that myeloid core of the granuloma, that center part, was dominated by a phenotype of macrophages expressing 2 proteins called IDO1 and PDL1 that largely suggest an immunosuppressive or a tolerogenic state. In addition to that, as I previously told you, most T cells get to the granuloma and they are blocked upon entry to the myeloid core. However, we found consistent with a tolerogenic environment that regulatory T cells or Tregs defied this rule set and instead were found to be infiltrating within that environment of macrophages expressing IDO1 and PDL1. Further supporting that this was a tolerogenic or suppressive environment and not a pro-inflammatory or antibacterial one, we found that TGF-beta and not interferon gamma was the predominant cytokine being produced in this environment. And this was a really important deviation from some existing paradigm in the field, and also led to a really adventurous time in peer review, which was that interferon gamma, which has long been associated to be the primary cytokine produced in response to TB, was actually found in scant quantities at both the mRNA and protein level. And instead, what we were seeing was the production of TGF-beta, a cytokine largely associated with immune suppression and wound healing. And we actually were able to even go one step further and look in the peripheral blood of TB patients, where we found this signature of what we called myeloid-mediated immune suppression was really emblematic of patients who had active or symptomatic TB disease. And so this study represented, I think a couple of really important shifts in the way we think about human TB granulomas as a disease process that is in fact setting up an environment to tolerate the organism, the MTB, and not clear it. However, there were a couple of questions this study left us with. First, why? What about the environment of the granuloma is imprinting immune cells with these features? What are the upstream drivers of things like IDO1, PDL1, TGF-beta? Why does the environment become compartmentalized in the way it is? And I think secondly, and probably even more importantly, we also weren't able to see any relationship between these features and the actual bacteria in the tissue. And this is simply because in human clinical specimens, we're not able to derive information about how much bacteria is in a particular granuloma. And so if you think back to that image I showed you in the first slide, where some granulomas sterilize while other granulomas are chock full of bacteria, this was simply not information we could derive in this study. And so with those 2 questions, we entered a sort of next generation of studies in collaboration with Joanne Flynn and Josh Matilla at the University of Pittsburgh. Joanne Flynn had pioneered a highly translational non-human primate model of TB that uses an organism known as the cynomolgus macaque. These animals, when infected with TB, recapitulate nearly all aspects of human TB disease process, including that diversity of granulomas. However, here, one of the really nice things about this system is that it's tractable. We know exactly the dose of infection the animals received. And importantly, we also can know information about how much bacteria is in a particular granuloma. And so with this, we put together a study where we took 16 animals representing 52 granulomas. These came from what we consider to be a critical window or inflection point of a disease at 9 to 12 weeks post-infection. This is where you see granulomas either start to do well and sterilize or do very poorly. As we wanted to look in this window as sort of that critical switch point to see if we could find the factors giving rise to that sort of splitting fate. We took these and we analyzed them with MIBITOF using an antibody panel that represented 38 proteins. Here we were actually able to scan much larger regions of the granuloma too, and that was a really important advance in the imaging technology that allowed us to generate images like you're seeing here, where we can look across the entire field of area of the granuloma and some of that surrounding uninvolved tissue. And from these samples, we evaluated the cellular makeup, the spatial patterning, the immunotopography, which I'll tell you about shortly. And as I mentioned, probably one of the most important features of this study is that for the first time, we were able to take all of the various features we can see by imaging and understand their relationship with bacterial control. And I'm showing here for each of the animals and each of the granulomas represented by the different dots or shapes on the screen here, what the CFU is. And what I hope you can appreciate is that in a single animal, you can have like NHP1 here. This animal has some granulomas that are doing quite poorly, but it actually has two granulomas, again, from the very same lobe of the lung that are successfully sterilizing. And so this cohort really for the first time has given us the chance to look at these very localized mechanisms of immune regulation that might help us understand why that particular granuloma is controlling infection while others are doing so poorly. And so I'm going to dive into a bit of the data. This has been a pretty massive data set, and there's a lot of different ways to look at it, but I wanted to walk you through sort of 3 vignettes of this data that I think have really helped us better understand the design principles of the granuloma that are leading to the immunosuppressive attributes we've seen, but I think that also highlight 3 different ways from a computational perspective of encoding and breaking down spatial features in data. And so I think, of course, I'm not sure anyone else in this room besides my own lab and maybe a couple others study TB granulomas, but I hope that as I walk through this, you can think about some of these spatial techniques that we had to either design or adapt for the setting of TB granulomas and how they might be relevant to other structures that have either radial symmetry or similar compartmentalization. And so I'm going to talk you through how we took this data and we're able to identify functional microenvironments, automate that process, and then start to understand the nature of these environments in the tissue. I'm going to talk to you a bit about an approach we developed called radial immunotopography to understand the sort of 2D radial distribution of both cell types and proteins in these tissues. And lastly, I'll tell you a bit about using cellular niche networks to really understand the difference between a good or controlling versus bad or permissive granuloma. But to go back to some very simple granuloma immunopathology, as I mentioned to you previously, one of the most characteristic and well-known features and probably long-standing features of TB granulomas are these canonical subzones of the granuloma. So I'm showing you here an example of H&E of one of our non-human primate tissues. And on the bottom, I've just overlaid what we would probably annotate as the 3 primary zones of this tissue. We have a central necrotic core. So this is comprised of cell death, some infiltrating fibroblasts. Actually, the TB does love to live in this environment despite being an intracellular pathogen. Next, we have that myeloid core environment. So this is that center cellular region full of macrophages and monocytes. And then in pink here, I'm showing the lymphocytic cuff. Here I'm showing you that same tissue from our MIBI data set, where I'm just showing you a few different markers, CD3 for T cells, calprotectin for neutrophils, and CD14 and CD11C for our macrophages, what these regions look like in our MIBI-TOF data. And so this is really our starting point. This is when you look at any sort of typical definition of a granuloma, you'll see these zones annotated. However, something that us and others have been observing is that there does seem to be, in fact, more than just this simple zonation of the granuloma. And this comes in the form of hypoxia. And so I'm showing you here work that was done by Josh Matilla at the University of Pittsburgh, where they were able to take some of these animals and infuse them with a small molecule known as pimonidazole, or PIMO, shown here in green, that actually labels hypoxic parts of the tissue in situ. And the nice thing about this is that you can label environments that are hypoxic in the tissue prior to necropsy of an animal, so you avoid any technical artifact. But the really nice thing about this molecule is you can also come in with an antibody-based staining assay, such as immunohistochemistry or flow cytometry, and actually visualize those regions after the fact. And so what I hope you can appreciate here is that if we look at this granuloma, we have this first part of the myeloid core that is predominated by the protein IDO1. This is the one I had told you we previously also identified as being quite significantly expressed by macrophages. However, in addition to that, there's this innermost environment that just lights up with pimonidazole, this hypoxia marker. And while if you look, if you take a step back, you might think these are all part of the myeloid core, But what we'd actually started to see is that this subzonation into both an IDO1 dominant and hypoxic dominant environment is a very consistent signature of TB granulomas. So in our study, we didn't have pimenidazole in all of the animals, but we did find that expression of GLUT1, which is a glucose transporter highly expressed during hypoxic metabolism, was a great surrogate for this environment. And I'm showing you an example here where in a different setting, you can see once again that same concentric organization, the myeloid core, where we have this outer region that's predominated by IDO1 and this inner region predominated by GLUT1. So using these two proteins, we actually went through every granuloma and we demarcated these zones using, just a very simple masking approach. And we redefined the myeloid core as actually being two environments, both the IDO zone and the hypoxic zone. And so you might be asking, why does this even matter? Why go through the trouble of dividing this environment? And the answer is because what we started to see when we went back and we looked at our cellular data is a couple of things. One, the biggest difference in granulomas that were able to control infection, so that's going to be those here in blue, that had a CFU, which is, here I'm using CFU, which is colony forming units as a surrogate for bacterial burden. So anything in blue means good, low bacteria, anything in red here means bad, high bacteria. We saw that really the only statistically significant differences between granulomas that were either controlling or non-controlling came from the macrophage compartment. And I'm showing you here, you can see that really the flavor of sort of macrophages that you find in a granuloma split almost along this line of whether or not a granuloma was controlling or non-controlling. Now, when we also took those same macrophages and we asked, where do they live in the myeloid core of the granuloma? Where do they fall? We found that the macrophages that were most enriched in granulomas that were poor controllers were also those found in the hypoxic environment. And so this was our first sign that perhaps this hypoxic environment in the granuloma is a key driver, or at least the primary niche of macrophages we know that associate with poor control of infection. I'm showing just a visual example here because I think pictures always tell a story better than bar plots, where I'm showing in one side here an example of one of our high burden granulomas where you can see these two concentric zones form. We have our IDO zone and our hypoxic zone. Here I'm showing you just what the distribution of macrophage phenotypes are. So you can see this environment has a real abundance of these CD11C macrophages, which were our most associated feature with bacterial control. And on the flip side, here's a granuloma, still necrotic, but one that has a much more diminished hypoxic environment and instead has an abundance of these CD14 positive macrophages or inflammatory monocytes. And so looking at this data, this began to suggest to us that hypoxia is not just a feature of the environment that's happening passively, say because of all of the cell death, but it's perhaps a microenvironmental feature that's preferencing and perhaps even imprinting macrophages with an immunosuppressive or a tolerogenic state. And so with this data, we wanted to understand if hypoxia, among these other metabolic features, had a greater influence on the entire design of the granuloma. Was hypoxia just there and it was really specifically influencing the state and organization of macrophages, or do these metabolic features actually seem to have some greater influence on the entire organization of the granuloma itself? And so for this, we turn to a technique that we call immunotopography. This was actually a really fun collaboration with Russ Butler at Adventist Health University, who works in the field of geographical information sciences. And so in a completely non-biological context, the field of GIS really attempts to take different types of map or satellite data and infer significant patterns of spatial organization. And I think this kind of speaks to the spirit of not reinventing the wheel, because while the field of GIS is not often concerned with looking at cells and tissues, what they are looking at, for example, are things like species and ecosystems, or the abundance of disease in different counties or zip codes. And so when you start to kind of take a step back and remove the names of the variables and just look at the structure of the analysis itself, many of the goals of GIS analysis are actually identical to what we want to do with spatial data. And so we've created a suite of different analytical tools that we're referring to as micro GIS. And I'm going to tell you about one of these today that we're calling immunotopography, which adapts an algorithm common in the GIS field known as buffer analysis. So the way buffer analysis works is we take a granuloma and we define the granuloma centers. Now in granulomas, these can come in two flavors. They can either be a solid center, this is when you have no necrosis or cell death, or they can be a necrotic center. I make that distinction because what we really want to look at is the cellular part of the granuloma. And so we define these centers. Once we define them, we perform geodesic expansion about these centers. And so what you can see here now is where I once had sort of this representation of nuclei in the granuloma and these centers. Now you can see there's these expanding, what we call buffer zones about each center. Once we have these buffer zones, we can extract any number of feature. We can extract the density of cells. We can extract the expression of different proteins. You can take any real representation of your data that you have, whether it be cell neighborhoods, cell abundances, and you can now encode those in a buffer-specific manner. And once we have that data, we can start to generate figures that look a lot like this, where I've taken one of my granulomas, I've overlaid the buffers, and now I can look at the radial distribution of cell types as I move from the granuloma center, or this margin between cellular inflammation and cell death, all the way out to the edge. And so this is a setting where, you know, an approach like this really applies quite perfectly. But I think if you have any kind of feature in your data that you want to anchor upon, say it's a vessel, say it's a tumor nest, say it's a duct, this kind of approach allows you to say, how do things move with respect to that anchored feature? So once we did this for each of the granulomas, we were able to actually integrate all of that information together, and this included both cell subsets and protein expression patterns, and produce a map like this, which we are considering our sort of consensus map of granuloma radial organization. And so across each of the columns here are these buffers. So this is from the inner part of the granuloma as you move to the outer part of the granuloma. And then each of the rows here represent one of these different features we measured. And so here, this analysis was restricted to both our cell densities and protein expression patterns. And so we can start to see here that certain features coordinate with each other, while all other features almost have complete mutual exclusivity with each other. And this is something that if we were to average across the whole granuloma, we're not able to see. But if we sort of move in this sliding window across the granuloma, these relationships start to fall out. Now, importantly, we could also take those automated zones that we had defined, the IDO1 zone and the hypoxic zone, and also overlay those here. And so now not only can we look at the relationship of features with each other over this space, we can also start to understand how they relate to other known pathological features of the tissue. And so this type of analysis has been extremely fruitful, and I think it sort of just like opened an entire box of different hypotheses and ideas, and really started to help us put together a model for why certain granulomas do well while others do poorly. And just to highlight a few of these features that come out, I'm going to show you both image examples, but also what we're calling these track plots. Where here, this is just looking at the feature density for the features that are shown in the image as you move across the granuloma. And so one thing that became very clear to us, and I think is probably the so-called smoking gun into why we think hypoxia is a pathologic feature of these tissues, is that T cells, particularly CD8 T cells, are almost allergic to this hypoxic environment. And this actually fits really nicely with what we know about the metabolic requirements of effector CD4 and CD8 T cells, where they really do require oxidative phosphorylation in a normoxic environment to enact most of their effector functions. And so you can almost think of this hypoxic environment as just a gate, you know, slamming shut on their ability not just to infiltrate, but also their ability to enact. Now, regulatory T cells, by contrast, which we had previously seen do successfully infiltrate the myeloid core, are actually much more metabolically permissive to different oxygen tensions. And a lot of this has to do with the fact that Tregs are found at a number of different mucosal sites in our body where they're critical for maintaining homeostasis. And many of those sites, for example, the gut lamina propria, can naturally have diminished oxygen abundance. And so the nice thing about this is that while this is a really important finding for us, it also biologically makes a lot of sense with what we know are the normal physiologic requirements of these immune cells. And so we see from this data that there's certainly this diminished T cell infiltration in the zone. We've also found that the IDO1 zone, despite being physically adjacent to that hypoxic zone, has an extremely distinct signature, both in the functional state of macrophages, and we think this is in large part being driven to the really unique epigenetic state we see these macrophages are experiencing. So here we're looking at IDO1 in blue, and then two different histone marks. One of these is a methylation on histone 3, and the other one is an acetylation. Broadly speaking, acetylations are usually indicative of active translation or transcription and open chromatin, while methylation is the opposite. And so it was quite striking to us that when we looked in this analysis, we see that you almost have a complete loss of the acetylation when you enter this IDO1 positive environment. And something that, when we've looked at this data, that's always struck us is that you have a very sudden change. You don't see sort of a gradient of expression of IDO1 or PDL1 in our prior data. It's all of a sudden you move 2 cells over and you're in this completely different environment of the granuloma. And there's a lot of, it's kind of mystifying to think about what microenvironmental cues could be driving such strict compartmentalization of a phenotype. And I think this suggests to us that this is in fact some epigenetic level regulation. We also see that cytokine gradients in the granuloma adopt a similar radial phenomena, where this IDO1 environment is characterized by significant evidence of type 1 interference signaling, something that others in the field have demonstrated is not a good thing for bacterial control. And lastly, we can also see that when you enter the hypoxic environment of the granuloma, you often see downregulation of HLA-DR, which is an important molecule for antigen presentation. And so this analysis has really allowed us to take what we were seeing in images, quantify it over the entirety of our cohort, and start to draw conclusions about how features within the granuloma environment might be influencing each other. Importantly, too, coming back to our metabolic attributes of the granuloma, we've also now been able to look at how features correlate across buffers as you move from areas that are more hypoxic to those that are more IDO1 dominated. And we can see, for example, that our immunosuppressive macrophages, these CD11C macrophages, have a positive correlation as you move from a buffer that is IDO1 expressing into a hypoxic environment, while consistent with the data I just showed you, the infiltration or presence of CD4 T cells drops off sharply as you move into hypoxic buffers. And so with this data, we've started, I think, put together a model for why we think the granuloma is adopting either an inflammatory or a tolerogenic state. But in the last bit of analysis I share with you today, I wanted to come back to what I said was probably the most important question we had in this study, which is what about bacterial control? So as I've mentioned, one of the greatest attributes of this study is that we can actually break down granulomas by how well they were controlling the infection itself, and that some of these granulomas are sterilizing infection while other are chock full of bacteria. And these granulomas actually fall over a 5 log-fold range of that variable. And so, the goal of this analysis is really to come back to those features, or what are things that we can think about targeting with drugs? What are things that we can think about incorporating into vaccine design? And so that requires us to look at bacterial control. And so I won't go, give too many spoilers on this, because I know Jolene, who's a member of the Angelo Lab, has developed probably what is everyone, at least here's new favorite spatial algorithm to look at what we call cellular niches or niche networks. But I wanted to show you what this data looks like when we look at these controlling or low burden versus permissive or high burden granulomas. What you're looking at here is a graph representation of the spatial relationships between cells in the granuloma. In this graph, each of these nodes represents a cell type. The size of that node represents how many spatial niches contain that cell type. And then the weight of the distance between nodes is inversely proportional to how often those cells co-occurred together. So for example, if we look at CD8 T cells, we can see that CD8 T cells were often co-occurring in spatial niches alongside these HLA-DR expressing lung epithelial cells. And so this is what the spatial network looks in aggregate for a controlling granuloma. We can see that it's a really densely connected network, that some of the really predominant players are CD8 T cells, these CD14 macrophages or inflammatory monocytes. But if we look at a poor controlling or a permissive granuloma, the network looks very different. Just at baseline, it's a much more sparsely connected network, and it's actually dominated, instead of this real diversity of different cell types dominating the niches, you actually see much more of a biasing towards these immunosuppressive macrophages. It's CD4 T cells. We actually see CD8 T cells, which we think are a really important driver of control of infection, are just out here, almost completely unrepresented in the data. Importantly, one thing we saw in our analysis was that endothelial cells or vasculature by composition didn't differ between these two settings. And you would think that if we're thinking about hypoxia, vasculature is an important feature for providing oxygen to the environment. But when we look at the niche network data, we actually see that while the magnitude or the presence of vessels was not different, where those vessels are located does appear to be different in these two settings, with the presence of endothelial cells much more interwoven among other types of both immune and non-immune cells. And so in summary, I've taken you through a few different ways that we've taken this data set, encoded different spatial features, and used them to really start to get a better understanding of why granulomas do well in some cases and not others, and what are the features that really drive that. And we've started to put together a model for hypoxia-driven immune tolerance in TB granulomas, where we really are referring to this as a so-called topographical trap, where lymphocytes, when they're recruited to the site of the granuloma, in some cases are encountering this almost impossible uphill battle driven by a diminished oxygen tension, resulting in hypoxia that seems to favor the presence of both regulatory T cells and macrophages that have this tumor-associated macrophage-like suppressive phenotype. And so I'm going to go ahead and stop there. Thank the really big team both at Stanford, and we have a huge network of collaborators who've worked together on this project. And as Mike mentioned, I recently started my lab where I'm continuing studies like this and others with the Spatial Immunology Unit. And so thank you to my team who are also here for their first conference ever. And with that, I would love to take any questions.
Any questions? Mike here. Got maybe time for a couple.
Don't be afraid, because I also would love to not take questions and go drink my coffee. But you can ask me a question.
Got so many questions.
Mike.
So, are you planning on making what infrastructure is needed if somebody wanted to use the GIS tools that you've put together here? Because I know you guys use like a mix of commercial software. Is it going to be something that sort of you think is deployable easily by other labs? Or
yeah, we're Pythonifying it, and so it was all originally done in this software that is not open source called ArcGIS, but now there's a really nice suite of Python tools, and so we're working on putting out a Pythonified version of 4 of these tasks. I showed you just one of them, but there's sort of 4 others that allow you to analyze the shape and the continuity of different structures in the granuloma. One that's very cool I didn't show here allows you to basically do a more sophisticated version of 2D cross-correlation analysis. And so that will be put together hopefully in the next year and published. Because I do recommend when you have a spatial analysis idea, going and looking to see if people in the fields of ecology, GIS, or fungal biology have done it.
So with the concentric ring analysis, right, you see a lot of organization with your kind of progression away from the core with the hypoxic and IDO zones, right? Yeah. But there's actually a lot more organization still as you go out. What do you think? Can you speculate a little bit, or what you want to look at about what could be driving that further organization?
Yeah, so I didn't get to talk about this too much. If you go to the very outer part of the granuloma where it looks like normal lung, it's actually not normal at all in most cases. And we see almost a second wall of inflammation. These lung epithelial cells that have HLA-DR upregulated, which is not normal. They're expressing interferon gamma. That's actually where we start to see what would resemble the inflammatory response we think we want in the middle of the granuloma. And so I think looking at some of these non-immune cell subsets, fibroblast populations, and then certainly the vasculature architecture, not to skip over the whole T cell part, although I would love to skip over the whole T cell section of the granuloma, but I actually think it's almost looking at that outer part that sandwiches in the pathology is where I would look next.
It's great to see the gradient from hypoxia and IDO. I think, do you suspect there's additional other gradients? Maybe other metabolic features kind of correlate with those primary ones, but they may have independent contribution to this type of a distribution.
Yes, definitely. And so Sarah, my postdoc here, is doing some work using IBEX, a multiplex fluorescence platform, actually with work that Felix Hartmann, who's going to be presenting this week, developed to actually profile metabolic state across all major metabolic pathways in these tissues. And we're pairing that also with spatial transcriptomics, where we can get a lot more sort of features of metabolic pathways. Because the short answer is yes, we do think there's actually a lot of complexity with lipid metabolism, iron metabolism, production of ROS. And so that's what we're currently working on now.
Hi, fantastic talk. I had sort of a chicken or the egg kind of question, sort of like what creates the hypoxic zone in the first place? Like, is it something the bacteria is causing? Is it related to like the size of the granuloma? You mentioned like vasculature, or is it like the hypoxic environment like somehow comes first and then alters sort of the state or the differentiation of the macrophages?
No, it's a great question. Exactly what we're trying to understand. One thing we do know is that the extent of necrosis, cell death in the middle, certainly correlates with the size of the hypoxic zone. It's not a perfect correlation. There are edge cases. We're doing a couple things to understand this. One, we're looking earlier. We're looking at a 4 to 6 week time point right after infection before we think this is established. And then, also there are cytokine gradients, even within necrosis. You actually do have viable cells infiltrating in that environment. So I think to understand it, we need to, 1, look earlier, 2, look in that environment, and then 3, actually, for the first time, I think I've been convinced to do some 3-dimensional imaging that I know Jonathan Liu will tell us about, to actually look at the so-called plumbing of the granuloma, see, what is the difference being driven in the presence of the vessels, the type of the vessels, the shape of the vessels, because I think probably that is going to also have a pretty large effect on the extent of hypoxia. And so that's a great question.
Awesome. Thank you.
All right. Thank you, Erin.
Thank you.
We're going to keep it moving here. And as Erin alluded to, our next speaker is from University of Washington. It's Jonathan Liu, and his lab, among other things, has developed, I think, a very exciting sort of extensible technology for being able to do 3D tissue imaging and whole FFPE blocks. And so he's going to talk to us about some of that stuff now.
All right, thank you. First of all, that was a great talk, Erin. Hard act to follow, beautiful work. It's great to be here. Thank you for the invitation to speak. It's always a pleasure to be back here at Stanford. It's been about a decade of my life here, the entirety of my 20s, so it has a special place in my heart always. So I'll be talking about non-destructive 3D pathology and analysis today at the University of Washington. I do need to disclose the technology I'll talk about has been commercialized by Alpenglow Biosciences, one of the sponsors for this conference. A company was started by myself and a former postdoc in my group, Adam Glazer, as well as 2 pathologists at the University of Washington, Nick Reeder, who's now the CEO of the company, and Larry True. Nick was a first-year resident when he started working with my lab when I first joined about 10 years ago at the University of Washington. Since Nick and Larry are both experts in prostate cancer, a lot of our initial work has been in prostate cancer. And so for people who have heard my talk, you'll know that I like to start with a bit of a case study to describe the type of patient that we're trying to help. So prostate cancer, of course, a very common disease, the most common cancer for men. And so men of a certain age were recommended to have our PSA levels checked. If those levels are elevated or if we have symptoms of prostate cancer, of course, then we go to see a urologist. And in the US, the urologist will take at least a dozen core needle biopsies. These are 1 millimeter diameter cylindrical plugs of tissue. If the pathologist looks at the slides that are generated from the biopsy and they see prostate cancer, one of the most important steps next is to determine the grade because most prostate cancers are relatively indolent. They don't lead to death, but there's a small fraction of prostate cancers that are aggressive that we need to treat aggressively. So about 50 years ago, Don Gleason developed Gleason grading. It's become the standard of care. And what they're looking at are relatively large architectural structures. So Gleason grading is entirely based on the glandular architecture. They're not looking at cytology. They're not looking at nuclei. They're looking at these relatively large glands that extend through the prostate and trying to score the aggressiveness of the cancer in that way. This is subjective. There's disagreements, as there is with almost every pathologist determination. And these disagreements can have major implications. For example, if it's entirely Gleason pattern 3, usually the recommendation would be to have active surveillance where the patient will come back annually for additional biopsies. This maintains the high quality of life, but of course there is a risk of metastasis if this is indeed aggressive cancer. If there's even a slight hint of Gleason pattern 4, usually that kicks it up to a very different treatment, surgery or radiation, which of course reduces that risk of metastasis, but has potentially serious side effects, incontinence, impotence. So this is a difficult decision for patients, obviously, as well as for urologists. And of the over 250,000 men diagnosed with prostate cancer each year in the US, roughly half of them lie right at this boundary between Gleason 3 and 4. It's a very common difficult decision. So in summary, Gleason grading works quite well, but it's not always reliable for distinguishing between lethal and indolent prostate cancer. And it's not just when the pathologists disagree, of course. Even if they agree, this is an approximate guide, and we should be able to do better. So one of the limitations is the way pathology is done. We all know how that's done, FFPE sections. Just a reminder, this is destructive. We're destroying the sample as we're sectioning through it. We like to be able to maintain the specimens for all kinds of molecular assays, of course. And we're only looking at perhaps 1% of the biopsy. And for a surgical specimen, much less than 1% is actually viewed by the pathologist. And we're looking at things in 2D. And so we'll talk about the limitations there. Of course, now we have whole slide imaging, but that adds another step with more time and money. We still have to go through this entire process and then scan the slide to do AI on it. So if we were to develop a process now, it might look more like this. We would want to fix the tissues, preferably without formalin, so we can preserve the proteins, nucleic acids better. Unfortunately, formalin fixation is going to be with us at least for the next few decades, if not century. It's hard to change that. But right now with FFP tissues, we can clear them quite effectively. We'll talk about that. Label them with fluorescent analogs of H&E or with antibodies, image it in a high throughput manner in 3D. This is entirely non-destructive of the specimen. We can image 100% of the biopsy in this case in 3D. And on top of that, it's easily automated. So it's non-destructive. So it's essentially a series of reagent exchanges that can be easily automated in an autostainer. So no histotech needed, potentially much cheaper and easier to achieve. So with pathologists that live in a 2D world, one of the questions always is, what is the value of 3D? It's sort of obvious to engineers and biologists, but clinically, this is not something that they're used to thinking about. Of course, we talked about convoluted branching tree networks like the prostate glands. There's other examples, tumor vasculature, kidney tubules, where you like to be able to see that in 3D. There's a lot of ambiguities. We'll talk about some ambiguities on 2D sections. Complex distributions of cells, the best example, as was alluded to, tumor immune microenvironment. These are highly heterogeneous structures and cell distributions. It often depends where you're looking. For example, you might want to be at the peritumoral edge, the infiltrating edge of the tumor, to see if the lymphocytes are trafficking in to predict immuno-oncology response. And then finally, sparse or rare objects, things like lymphovascular invasion, perineural invasion. We know they're there. Pathologists can see them on 2D sections, but they're often missing them. If we can image orders of magnitude more tissue and apply AI to find these rare events, of course, we can quantify these structures better and improve patient decisions. So we use light sheet microscopy. There's two main advantages of light sheet microscopy. One is compared to confocal or two photon, which are laser scan techniques, where you have to generate the image point by point. Here, we're using a fast scientific CMOS camera to collect millions of pixels of data all at once. And if we scan the specimen through the light sheet, we can reconstruct the 3D volume very quickly. It's also very gentle. Instead of cutting the tissue into a thin tissue section, we're illuminating the tissue with a thin sheet of light. And so we're only sending light to the part that we want to image. So it's much more optically efficient than confocal microscopy, much less photo damage, much less phototoxicity. So in the early days, this was about 20 years ago, they started using this for developmental biology, looking at these relatively transparent model organisms, embryos, Drosophila, zebrafish, where the low phototoxicity was a major advantage and the speed as well. Now for clinical applications, light sheet microscopy traditionally was not ideal because you had these bulky objectives arranged orthogonally. The sample prep was very tedious, agarose embedding, mounting the specimen on the tip of a cuvette or a capillary tube. And so it would not work for a high throughput clinical setting where you have multiple specimens of different sizes. And so in the early days, we decided to build essentially a flatbed document scanner for tissues. All of the optics are underneath a modular sample holder. It could be a glass plate, a multi-well plate, or a custom biopsy holder. Here you can see with our first generation device, the light sheet comes in at a 45-degree angle. illuminates the tissue, and then we image orthogonally onto our scientific CMOS cameras. A lot of the challenges and a lot of the IP that we have has to do with how do we get the light focus into the tissue through these sharp interfaces at such a strong angle. So these off-axis aberrations are quite severe unless we play some tricks. The light's going from air into glass, into tissues, different refractive indices. So that's challenging, and we've detailed that, of course, in our publications. This is a stage scan system, meaning the light sheet is stationary in space. To generate a 3D data set, we move the tissue. These are fixed, clear tissues that we move. For example, if we move left to right, we collect a series of these 45-degree angled light sheet images that gives us a volumetric brick of data. And then we can scan adjacent bricks of data to scan a very large volume over time. So again, the open top gives us that ease of use. The light sheet provides the speed. So here we're showing our first 4 generations of OTLS microscopes. Over the years, we've tried to both improve the resolution as well as ruin the resolution, so to speak. And our last 2 systems have been multi-resolution systems. So if you think about how a pathologist looks at a glass slide, most of the time they're at relatively low resolution using 2x, 5x, or 10x objectives. Only rarely do they zoom in with the 20x or 40x magnification objective. That's even more important in 3D. The data set sizes, the imaging time scale to the 3rd power with resolution. So this multi-resolution workflow becomes really essential for larger specimens. Our latest system also has the largest working distance of 1 centimeter, so we can image whole mouse brains, for example. And then this is a bit of a subtle point, the tolerance to index mismatch. So with our first 3 systems, as we improved the resolution, we became more sensitive to the index matching of the specimen and the specimen holder. In order to mitigate aberrations, we had to match those materials quite precisely. With our latest system, we've figured out a way to get around that, to get around those off-axis aberrations. So now it's a much more user-friendly system. We're not as sensitive to matching the index of the sample and sample holder. Just have one slide on tissue clearing. We use most of the time iDisco, which was actually invented by the former president of Stanford, Mark Tessier-Levine, back when he was at the Rockefeller. A lot of people use this because it's very, very simple. It's basically dehydrating the tissue. Water is really the biggest culprit. It has an index of 1.33, which is quite low compared to the lipids and proteins in the tissue. So if we use ethanol and we dehydrate the tissue and replace the water with a high index oil, in this case, we use a food-grade cinnamon oil, ethyl cinnamate, at an index of 1.56. Most tissues become quite clear, up to about 0.5 millimeter, 500 microns deep. We can image. You can also image bone. There's ways to decalcify. Pretty much anything can be cleared these days. As you can see, these images look like H&E, and that's intentional. Of course, that's what pathologists are used to seeing. So in the early days, we've worked hard to develop a fluorescent analog of H&E staining so that the images, we can convince the pathologist we have the resolution, we have the image quality, it looks identical to a whole slide image. There are some tissues that are more challenging to clear. Also for fluorescent proteins in preclinical models, they tend to bleach out with the ethanol dehydration. So there are aqueous protocols that we've used. Cubic is a great one, where they delipidate and they have other tricks to keep things in an aqueous environment. This works also better for certain antibodies that don't want to be in dehydrated conditions. I should mention, the false coloring here is done digitally, of course. Our fluorescence microscopes use grayscale black and white cameras. We use, of course, fluorescence filters, but the data sets are grayscale. A lot of people are using deep learning now for image style transforms, image-to-image translation. We use a physics-based model of the Beer-Lambert law absorption of light to do this. No hallucinations, very deterministic. And that's because we stain with a fluorescent analog of hematoxylin, a nuclear fluorophore, as well as eosin happens to fluoresce already. And so we can apply this Beer-Lambert law inversion to make it look like H&E. Here's just an example of a preclinical data set. This is mouse kidney stained with a variety of labels for nuclei, for collagen, for the vessels. Here's the glomerulus poking out here. This just gives you an example of the richness of a 3D data set. in this case, prepared by Josh Vaughn's lab at the University of Washington using hydrogel embedding and tissue expansion. Here you can see some of the white blood cells and red blood cells floating in space. Here's another nice data set illustrating a multi-resolution workflow. Here during the pandemic, the inventors of Cubic mailed us these whole mouse brains that they had cleared. And so they were studying brain metastases. We first needed to image the whole mouse brain to find the location of the mets. And then we can zoom in with the high resolution mode of our microscope to really quantify these structures. If we were to image the whole mouse brain at high resolution, it would have taken us a week. With the low resolution mode, 4-micron resolution, we could do that in a couple hours. So it really tells you why we need this multi-resolution workflow. And then finally, going back to prostate, this was actually acquired with our second generation system. This is a 1 millimeter diameter needle core. We're going from one end to the other. Staying with our H&E analog, you can start to see, in this case, benign prostate, this branching tree network of glands that's so important for grading the severity or the aggressiveness of the tumor. For cancer, one of the biggest ambiguities is distinguishing between a variant of Gleason pattern 4, which is called a poorly formed gland, versus they know that if it's a fully formed gland and you cut it tangentially at the edge, it will look poorly formed. So this is an ongoing ambiguity that they always struggle with. Here's just an example where at one depth, you see this collection of epithelial cells. It lacks a lumen, it looks like a poorly formed gland. But clearly, as you go deeper, you see it's part of a fully formed gland. So as a reminder, this could be the difference between a patient needing to have surgery or radiation versus active surveillance. So this is important. There are many examples like this in pathology. For example, we're imaging tumor buds in colorectal cancer specimens, same thing. Things that look like isolated tumor buds in 2D often turn out to be infiltrating tendrils emanating from the main tumor mass in 3D. So that kind of gives you an example of why 3D can help. Of course, the better way to analyze things is with machine learning. So a few years back, we developed a pipeline to segment out the glands. Of course, the glands are the best place to start because that's what the pathologists are already using for Gleason grading. So we can look at the lumen spaces in red, the epithelial spaces in yellow, and then the skeleton network was the magenta branching tree network. Right here. So then we did a very preliminary study with 50 cases. Half of these patients had recurrence of their cancer within 5 years of prostatectomy, and half of these patients did not. So that's our high-risk group and our low-risk group. We took simulated biopsies from the prostatectomy specimens. So we extracted 6 biopsies from the 6 regions that urologists usually target for their biopsy procedures. This is all done ex vivo. We stained it with our H&E analog, cleared the tissue, did our 3D microscopy and gland segmentation. And then really the big question was, let's compare 3D versus 2D glandular features. So if we looked at volume ratios in 3D, we looked at area ratios in 2D. If we looked at curvature in 3D, we looked at circumferential curvature in 2D. We wanted to compare side by side the value of 3D versus in 2D, we looked at 3 different levels to replicate the standard of care. Usually pathologists will look at 3 cross sections separated by about 20 microns. And then we correlate these features, glandular features, with the biochemical recurrence outcomes, the 5-year BCR outcomes for these patients. So here's an ROC curve where we took about a dozen features, glandular features, not a huge amount. And we looked at the 3D versus the 2D version, where clearly the 3D is more predictive of the risk of the patient. And for some of these patients, we had time to recurrence. So 0 is the surgery date. Over the course of 5 years, every time you see a drop, that means one of the patients had a biochemical recurrence event. So this is a progression-free survival, BCR-free survival on the y-axis. And so the point here is with our best 2D model, we don't see very good stratification between high-risk and low-risk groups. But with the 3D model, we can predict who's going to have recurrence. So far, we've talked about this sort of handcrafted approach where we can first segment out the structures that pathologists trust, glands, it could be nuclei, it could be other things. And then we extract very intuitive features to train a more traditional machine classifier. Alternatively, we know we're missing things. There's all kinds of subvisual features, nuclei, vessels, nerves. we could do this black box end-to-end deep learning approach. And so that's something we've done in collaboration with Faisal Mahmood's lab at Harvard, and he'll be speaking tomorrow. So there's really three main steps to this pipeline. First, we have to divide the biopsy into chunks to fit into the GPUs. Then we have to extract features. There's no foundation model for 3D pathology yet. And so they used a variety of radiology models and also video models where you treat the time axis as the z-axis. Turns out that worked the best. And then once you have a low dimensional feature representation for each chunk, you can aggregate all the chunks within the biopsy and try to train things to predict, in this case, the BCR outcomes of the patient. So this was a very long paper. I think it's all summarized in this one bar plot, really. Here we have a prediction of the BCR outcomes of the patient based on the original pathology report. So that's pretty good, and it's based on Gleason grading. But if we apply 2D AI to a few sections, to 3 levels within the biopsy, that gives us better performance. If we apply 2D AI to the entire biopsy, level by level, 2D plane at a time, that gives us better performance because we're looking at more data, the whole biopsy versus just 3 levels. But the best performance is when we extract 3D features. These are the features that we could never extract from a 2D image. They're out of plane, they're volumetric features, and that gives us the best performance. So there's really 2 advantages. One is more data, 2 is volumetric features. That gives you the best performance. And it turns out for these small biopsies, the more volume you look at, the better the performance is. And just one final story. What I talked about before is sort of a fully computational way to stratify patient risk, or maybe it's predicting treatment response in the future. Here, what we're trying to do is to find a more low-risk path towards rapid clinical adoption, where we keep the pathologist in the loop. And so with this pipeline, we wanted to extract out the highest risk levels to show to a pathologist, let them make the final diagnosis. Instead of just extracting random levels for them to review, we're trying to find the best levels using this pipeline. So to make a long story short, in the end, we did a very preliminary study with 20 biopsies. These are endoscopic biopsies from Barrett's esophagus patients where we're trying to screen for dysplasia or cancer, trying to look for neoplasia. At the University of Washington, they have to look at 16 image sections per biopsy, so quite a bit of work, and there's usually dozens of biopsies per patient. With the AI triage 3D pathology, we extracted, well, we first imaged the entire biopsy, but then we used AI to find the 3 best levels. And we found that with a traditional pathway, they only found 1 case, 1 biopsy with neoplasia. We were able to find 4 with the AI triage pipeline. So in summary, we can improve the detection of neoplasia while simultaneously reducing pathologist workloads. So that's kind of the win-win scenario. Pathologists are all overworked these days. It's a big problem with the workload. So let me just summarize. With all of our imaging techniques in this room, there's always the sample prep, there's the imaging and the data processing and analysis. We're working on all of those categories, as are many of you. In terms of tissue prep, we've worked hard to try to automate these things and make the quality control better. In the early days, we'd be happy if 10% of our samples looked good. Now, over 90% of them look good the first time. We've really refined the process, and we have a pretty rigorous QC process after we image our clinical specimens. In terms of spatial omics, of course, that's a hot topic, trying to push things into 3D. Although, these are 2 big data fields, bringing them together is just going to be this massive data problem, I would say, or challenge. But nonetheless, places like the Allen Institute in Seattle, they can do it. They have hundreds of people that are all focused on these types of issues. We're collaborating with them on some of these projects to push spatial transcriptomics into 3D. And then we have a poster today or tomorrow on 3D guided macro dissection, finding regions in our 3D data sets that have unique morphologies where we can macro dissect out and do whole genome sequencing on. So that'll be a poster from Hoi Ching tomorrow. On the imaging side, of course, people always want to improve the resolution and depth, including in fresh organoids or organ on chip. So we have a few projects on that. pushing things into neuroscience with Allen Institute, also traumatic brain injury. And then finally, this new project on surgical guidance, where we want to be able to image freshly excised lumpectomies or head and neck specimens, and to be able to spot positive margins within 15 minutes of excision. That's a really moonshot project that was recently funded by ARPA-H. And then on the computational side, of course, there's many opportunities, not just with glands. We've published on nuclear features with Rob Serafin in the lab. He has a poster on a pipeline to automatically diagnose glands. That's always the first step. We need to know which glands are cancer, which glands are benign before we do any other downstream analysis. And then Sarah in our lab is working on nerve and vessel segmentations to be able to see those features in our 3D data sets. And then finally, for end-to-end deep learning, Gan Gao has a poster trying to take our AI triage 3D pathology to the next step. The original pipeline was based on fully supervised learning, based on very tedious pixel level annotations. Now we're doing things in a more weakly supervised way, which should give us better training set diversity. So it should be better. And then finally, the end goal is really to integrate the AI with the imaging itself. These AI guided imaging pipelines for multi-resolution imaging, that's going to be a part of the ARPA-H project. So let me end there and just acknowledge different collaborators. Most of our funding's from the NIH and DoD, and recently ARPA-H. So yeah, thanks again for this opportunity.
Yeah, time for a couple questions.
Hi, so you spoke a bit about how the 3D kind of resolution or dimensionality is so much greater than 2D. So can you maybe give us a sense of, when you look at a 3D block, how many, sort of order of magnitude 2D slices would be in there? Is it in the tens, hundreds, or
yeah,
what's
it's 2 to 3 orders of magnitude larger. So we're looking at the equivalent of hundreds to thousands of 2D sections in our 3D data sets. So quite a bit larger. Yeah.
Jonathan, fantastic talk. I have 2 questions. The first one is on the 3D segmentation side. Do you need to introduce additional dyes to actually segment the glandular structure? If so, what's the maximum in terms of that you can introduce?
Yeah, so that's a good question. To train that gland segmentation algorithm, we did stain the epithelial cells with a cytokeratin marker. But then we trained a deep learning algorithm to replicate that stain, so synthetic labeling, we call it. And then that allowed us to start with just our H&E analog in the future and never have to do immunolabeling. And that's because with immunolabeling in thick tissues, as you all know, it takes time. In our lab, it took 3 weeks to do immunolabeling of thick tissues. So we wanted to, in this case, since these are relatively easy structures to spot based on H&E, we knew we could just train an algorithm based on just the H&E. The H&E is based on staining with a small molecule reagents that diffuse very quickly. We can do the H&E staining within a day, I would say.
Gotcha.
Yeah.
Sorry, the second question I want to ask is, you mentioned one of your 3D project with the H&E, you started with pixel level annotation. So what's your favorite 3D annotation tool for that?
I believe they use QPath. It changes monthly. Now we're using, this will be a good question for students. I think we're using more Napari now. And there may be some other tools that people are using, but that'll be a good discussion to have with others. So we're still trying to figure out, especially when we involve pathologists that are maybe not as used to some of these types of tools, trying to find the most user-friendly way for them to annotate structures. Yeah, that's something that we're actively trying to do.
Thank you.
Yeah, thank you, Professor Liu. And first question is that theoretically, I believe in the advantage of 3D technology over 2D, but the prostate cancer problem has been addressed by many researchers, and PageAI has been developed clinical grade AI models with FDA approval based on 2D images. So I'm curious, what actual benefit can we have from 3D images in clinical scenario? And another question is about the overfitting problem. So many of the study conclusion are based on like 100 samples. And we know that 2D images is gigapixel and 3D images is tens of gigapixel. And now we only have 100 of labels for these images. So how can we convince that the deep learning model do not overfit the data set? Yeah.
Yeah, those are good questions. And you sound like reviewer 3. No, those are.
Or you reviewer 3?
Yeah, those are the challenges for us in the future, and really the subject of our NIH grants as well. To scale things up, we have to look at more patients prospectively. Ideally, we have to prove more rigorously that 3D is better than 2D. And so, in the future studies, we really want to try to compare exactly with the standard of care with 2D pathology and with AI tools looking at 2D pathology. So far, we have a few studies suggesting that 3D analysis is better than 2D analysis. But like you mentioned, we need to make those studies larger and better. And so, yeah, that's for the future for sure. Yeah.
Okay, I think we're going to need to keep moving so we don't get too far behind. Thank you for that wonderful talk, Jonathan. Now, for our next talk, we have actually, I think this is our first trainee talk. We're going to have Noah Greenwald come and talk about some of the stuff that he's been doing looking at checkpoint response in breast cancer.
Great, thanks, Mike. Good morning, everyone. My name is Noah Greenwald, and I'm excited to tell you about our work profiling the response to immunotherapy in triple-negative breast cancer. Immunotherapy has fundamentally changed how cancer patients are treated across a variety of cancer types. Shown here is a Kaplan-Meier plot from the KEYNOTE-355 trial, where pembrolizumab, an immunotherapy targeting the PD-1 protein, was approved for advanced triple-negative breast cancer. So you can see the orange line here, which is the patients who were treated with immunotherapy, ends up higher in the bottom right-hand corner than the blue line, which is the patients who received placebo, indicating that these patients benefited more. This was a really significant advance and has already fundamentally changed how breast cancer patients are treated. However, there's still significant room for improvement. In particular, you can see that both of these lines still end up pretty low in the bottom right-hand corner, indicating that the majority of patients are actually not benefiting. In addition, the orange and blue curves are not that well separated from each other. This is despite the fact that PD-L1 positivity, which was used as a biomarker for this trial. This tells us that we still don't really understand what separates the patients who are going to respond from the patients who are not going to respond. To help fill in some of these gaps, we constructed a longitudinal cohort of triple-negative breast cancer patient samples. We collected the primary tumor from each of these patients. Following this, all these patients' cancers metastasized, at which point they were eligible to enroll on the TONIC clinical trial. We collected a metastatic baseline sample at the beginning of the trial, a metastatic pre-nivolumab sample following 2 weeks of induction chemotherapy to prime the immune system, and a metastatic on nivolumab sample following 3 cycles of anti-PD-1, which is the target of nivolumab. This gives us a really unique opportunity to understand the longitudinal evolution of the tumor microenvironment in these patient samples and how that evolution corresponds to the patients who did or did not receive benefit from the immunotherapy. To characterize these samples, we took advantage of an approach that our lab has developed, which Erin McCaffrey previously described, MIBI-TOF. This allows us to look at dozens of proteins at once in the same section. Here, I'm showing the markers that we included in our study, including a number of different immune lineage markers to identify the major immune cell types, checkpoint molecules, including PD-1 and PD-L1, tumor markers to identify the tumor cells, structural factors to allow us to identify extracellular matrix proteins and fibroblasts, as well as functional markers to describe the activation and regulation of these cells. We can then use this data to understand the spatial features of the tumor microenvironment. After generating multiplex imaging data with MIBI on all of the patient samples from each of the patients, the first task is to identify and classify the cells in our images. Here in this heat map, each row is a different cell type and each column is a marker that was used to define those cell types. Red indicates high expression of a given marker in a given cell type, and blue indicates low expression. We identified 4 major classes of cells, including cancer cells, lymphoid cells, myeloid cells, and structural cells. I think what you can appreciate here is that each row, which is a different cell type, tends to have a different set of markers that define it from all of the other rows. This is why it's so critical to be able to look at dozens of different proteins at once, so we can really capture and describe the full diversity of cells that are present in these samples. Once we had identified the location and the cell type of every cell in every image, we next needed to extract the key information from this image data. When we started this project, there was not a single unified way that would allow us to really comprehensively describe all of the features in our images. To address this gap, we created an algorithm called SpaceCat, which generates a spatial catalog of image features. It gives us a really comprehensive description of all of the key components of our image data. Here, I'll walk you through how this process works. One category of features that we're really interested in is cell abundance. Here, we've colored all of the CD8 T cells in yellow. The top image has a really high density of CD8 T cells, and the bottom image has a very low density. We can do this density calculation separately for each of the cell types we identified across all of the images in our data set. I mentioned before that we also included a number of functional markers on our panel. Here, we're showing Ki67, a functional marker we use to identify proliferating cells. We've colored all the Ki67 positive cells in red and all the Ki67 negative cells in gray. The top image has a really high proportion of Ki67 positivity, whereas the bottom image has a very low proportion. We can do this separately for each of the functional markers in our panel, across each of the cell types we identified, separately for each of the images in our data set. Moving beyond individual cell type abundance or phenotype, we can also look more holistically at the overall composition of the images. One way we do this is using the Shannon Diversity Index. This allows us to evaluate the relative balance or imbalance in proportion of cell types in a given image. Here, we've colored each of the cell types with a different color. The top image has really a large number of different colors, indicating a high diversity of cell types, whereas the bottom image is composed almost exclusively of a single cell type, indicating low diversity. We can do this at different levels of granularity across all of the images in our data set to generate a comprehensive set of diversity metrics. So this is the output of SpaceCat. It basically allows us to automatically quantify a large number of distinct features to summarize the key components of image data. Our first question then, once we had generated these features, is would they allow us to understand what separates the responders from the non-responders? Here I'm showing a volcano plot where the x-axis is the strength of the association with response, and the y-axis is the significance. Each dot here is a different one of those SpaceCat features. What we found is that there were a large number of features that really robustly predicted which patients were going to respond and which patients were not going to respond. We were curious if there were any patterns in the types of features that were best able to separate responders and non-responders. One thing that we noticed right away is when we looked at the features that required spatial information to be computed, those were enriched in the most predictive features compared to those that did not require spatial information, sort of validating this approach and that we are extracting unique information with our spatial data. I mentioned previously that we collected longitudinal samples from these patients. We were curious then how the features we identified changed as we moved through time. Here we're looking first just at the primary tumors, and we've colored each of the features in dark blue if they were robust predictors of response. Looking in the primary samples, we identified only a single feature that was able to predict which patients responded and which patients did not. When we look at the baseline and the pre-nivolumab metastatic samples, we now see a greater number of features that are associated with the response, but it's really the on-treatment sample where we have the most information about which patients are going to go on to respond and which patients will not. One thing to note here is while there are some features that seem to be important and are shared across multiple time points, there are other features which are unique to a single time point and only predict response at one or two of these different times. Everything I've showed you so far has been looking at individual features in isolation from one another. These differences we saw across different time points, though, prompted us to wonder if we could integrate all of the information from these different features together, how would our ability to predict which patients responded or did not respond change? To answer this question, we constructed multivariate models using all of the features from a given time point. Here we're using AUC as a measure of accuracy, where 0.5 indicates essentially random predictive performance and 1 would indicate perfect performance. Looking just at the MIBI data from the primary samples, we see that multivariate models do not do a very good job of predicting which patients are going to respond and which patients are not. However, when we look at the baseline and pre-nivolumab metastatic samples, we now see that especially the baseline sample does a much better job of separating the responders from non-responders with an AUC of 0.77. Interestingly, this is the exact same AUC that was recently reported from another spatial analysis of triple-negative breast cancer patients who were enrolled in a clinical trial for immunotherapy. The difference with this other data set, however, is that these patients were only had primary tumors that were being treated. One thing to note is from this previous publication, they had the exact same AUC, 0.77, for the pre-nivolumab, for models trained on the pre-nivolumab samples and models trained on the on-treatment samples. In our data, we saw something very different, where the models trained on the on-nivo samples had an AUC of 0.9. So we were able to do a much better job of separating which patients responded from which patients did not. In addition to the MIBI data, we also had bulk RNA sequencing data from a subset of these time points. We did the same exercise, training a multivariate model based on this data, and found something pretty striking, where the on-nivo sample, we saw really similar performance between these two modalities. However, looking at the earlier, more challenging time points, we see that the MIBI models, models trained on the MIBI data do a better job. We also had bulk whole exome DNA sequencing data from one of these time points, and we could similarly do the same exercise. However, we found that this data really did not capture the key information that allowed us to separate responders and non-responders. In summary, we profiled 117 triple-negative breast cancer patients through treatment. We identified spatial and non-spatial determinants of response, looking at these SpaceCat features, which allowed us to generate this really comprehensive description. When we looked at these predictive features, we found that they varied over time, with relatively little information in the primary tumor about which patients were going to respond, and the most information on the metastatic, and particularly the on-treatment samples. We also saw different predictive power based on the modality, with the MIBI and RNA-based models doing a much better job than the DNA-based models at predicting response. Wanted to thank everyone who is involved in this project, in particular Iris Netterlorf, who co-led this project with, everyone in the Angelo, Koch, Corlings, and Curtis labs for their support, my funding, you all for listening, and we actually just put out our beta release of SpaceCat an hour ago, so if you're interested, just a coincidence, if you're interested in trying it out on your data, please check out the GitHub link and let us know what you think, and I'd be happy to take any questions. Thank you.
Okay, any questions for Noah here?
Thank you, Noah, for a fantastic talk. I'm just curious of all the features. If you were a physician to pick how to go, for clinical translation, which ones would you do? And would you still start from multiplex imaging and then, doing all this analysis, or would you just say pick a few and just do a quick stain? What is the approach to go forward for clinical translation?
Yeah, that's a great question and something we've been thinking about as well. It's definitely not the case that you need the full breadth of MIBI data to generate predictive models. We actually looked under the hood, and these models, because of how we designed them, were relatively sparse. So it's only 5, 6, 7 features that are really driving the performance. Those depending on the time point, 15 to 20 markers to get the full performance. We think you could go maybe even half of that and still rescue a lot of it. So I think it really depends on the specific question, which of those, potentially 37 proteins on the panel you would most want to prioritize and do a much smaller number that I think would be more feasible to translate.
Really nice. I'm wondering about the features that you identified only at certain time points as being relevant? Did you look into what these features really were and whether they're indicative of dynamics or if it's really about a process that is only visible at a given time?
Yeah, great question. So I think one pattern we saw that surprised us was that the lymphocyte-based features, which I think, as Erin alluded to, everyone is focusing on T cells. We saw that those were really only important for the on-treatment time point, and that before administration of the drug, it was not these T cell-based features that were predictive, but it was more structural macrophage, ECM features that were coming up. So there was a little bit of a bifurcation there where, you know, the readout of patients that were responding well was infiltration of a diverse array of different lymphocytes, but the pre-existing lymphocytes, at least in the metastatic tissues we looked at, was not a strong predictor.
Thank you.
Hi, great talk. I was just curious in your modeling approach, if you could speak to how you guys deal with collinearity and spatial features that are dependent on each other.
Yeah, so we took a two-pronged approach to that. One is that within SpaceCat, there's a correlation cutoff. So we have sort of a hierarchy of which features we think are more important and potentially less important. And if any of the less important features are very highly correlated within sort of the same branch of more important feature, we don't include it because it's basically redundant. We also use, in this case, a lasso, which is like robust to that type of correlation, but you will end up arbitrarily picking one of those correlated features. So it doesn't impact your performance, but it can impact your interpretation if you have a lot of correlated features, because somewhat arbitrarily one of them will be included in the model.
Great, thank you.
Thank you, Noah.